Adventures in Brain Injury podcast series. Today's episode is sponsored by Minnesota Functional Neurology and Chiropractic, found online at mnfunctionalneurology.com. Our thanks to Dr. Jeremy Schmo and his team in Minnesota. And our thanks to Revive Treatment Centers of America, found online at revivecenters.com. Dr. Joshua Flowers and his team in Colorado. We send our thanks for your sponsorship today.
Absolutely, and I want to give a shout out to Dr. Jeremy Schmo over at Minnesota Functional Neurology and Chiropractic, and to Amy Zelmer for launching the Brain Health Online Summit, which I'm actually a presenter on. So me and so many great practitioners, some of which who have been on the show, including Dr. David Traster, including Jeremy Schmo, of course, and Debbie Zielinski, who is going to be on this show soon. She's a neurooptometrist. There's all sorts of really great people. Christabel Braden is going to be on. So check that out. It's really bringing safe and effective methods and modalities for brain injury rehabilitation to the forefront. So check that out. It's thebrainhealthonlinesummit.com. Also, I want to announce that I am taking consultations. If you go to feedabrain.com/consultations or adventuresinbraininjury.com/consultations, you can schedule a free 30-minute consultation with me. So, Michelle, how excited are you about this episode?
Oh, Calvin. It is a very precious opportunity to speak with a budding neuroscience researcher on fresh, novel neuroscience research. We are so fortunate.
I was about to say, aren't we lucky?
Oh, beyond lucky.
So let's get into it.
Let's go.
All right. Hello, everybody, and welcome to the 24th episode of the Adventures in Brain Injury podcast. My name is Calvin Ballister. I'm a severe traumatic brain injury survivor, creator of AdventuresInBrainInjury.com, and creator of feedabrain.com. And with me, as usual, is Michelle Malmberg, acquired brain injury survivor and co-host of the Adventures in Brain Injury podcast, and amazingly lovely human being. How are you doing, Michelle?
Thank you, Calvin. I'm doing very well today. Yeah, I'm really excited to speak with our guest this morning.
Me too. Who do we have with us?
Oh, we have a beautiful soul. Her name is Ms. Indrani Das, and I am not going to, there's going to be no fluff here. I'm going to dive straight in. We've got a little surprise for everyone. So I want to tell our audience, our listeners, just a little bit about Miss Indrani Das, who recently won the 2017 Regeneron Science Talent Search and a small prize of $250,000. This competition is one of the nation's oldest and most prestigious student science and mathematics competitions in the country. And we are so fortunate to have her with us. She's in the lab right now, hopping between incubators, test tubes, centrifuges, and microscopes, and we get her for the morning. So there are some surprising details about this researcher we are going to announce later, but we're going to keep you all hanging for now and listening to the rest of the podcast. Right now, Calvin and I would like to ask Indrani to take us on a tour of what she sees through the lens of her microscope and what happens to the brain on a microscopic level after injury. What do you think about that, Calvin?
I think that's a great call. Indrani, welcome to the show.
Thank you. I'm so excited to be here.
Yes.
We are equally excited to have you, maybe even more.
No, it's incredible. I've been chatting with you a bit, and it's just like I've been blown away by your research and your novel treatment on brain injury, which we are going to be speaking about today. And as Michelle said, why don't we start and just dive right in and take us on a tour of what happens to the brain after injury and what we see at a cellular level.
Of course. So the first thing I'd like to say is that brain injury isn't really what everyone thinks it is. A brain injury on the most basic level is some kind of an insult to the cells of the brain. So this could be a blunt force to the head, a traumatic brain injury, or other types of diseases such as Alzheimer's or Parkinson's, where there are proteins kind of like clogging up and causing this injury to the cells around them. So on a basic level, brain injury is when the main cells of the brain, called neurons, receive some sort of insult. And people don't often know what goes on around this. There's a whole nother system of cells in the brain besides these neurons. They're called glial cells. And these glial cells make the main response.
Before you go into that, I just want to thank you for explaining how a brain injury is far more than bumping your head or smashing your head. Like a brain, I remember watching some presentation that says a brain injury, a mild brain injury, traumatic brain injury can be caused from a roller coaster. And what we're also, yeah, it's great. I mean, but I definitely appreciate looking at the brain disease and any insult to a brain.
It sets off a whole chain of events.
It does, and that's a brain injury. Brain injury is neurodegenerative disease. It is Alzheimer's. It is dementia.
Stroke.
It is strokes. It's definitely strokes. I suffered a hypoxic stroke while I was in the ICU after my injury, and so sorry. Just wanted to thank you for explaining that.
Well, and what about? I think overlooked are the everyday concussions, the chemical concussions that people inflict upon themselves with their food choices.
Yeah, absolutely. Especially with with so many new compounds coming out and being added to our foods, like MSG, monosodium glutamate, which we'll be talking about glutamate quite a bit. And other free glutamates that aren't labeled as MSG.
And sugars, just high blood sugars.
There you go. Yeah. Well. Why don't you tell us, Indrani? Why don't you tell us about glia? That's what you were getting into.
Yes.
Right. So basically, that whole chain of events that happens after there's some kind of damage to a neuron. What these neurons do is they send signals to these glial cells around them saying, help, I'm injured. And these glial cells basically rush to the area. They surround these neurons. They kind of try to keep the damage to one area. And for a while, this response is helpful. And it's aimed towards kind of restoring health to these neurons. But if this process becomes really prolonged or severe, by the way, it's called gliosis. I worked with a specific type of gliosis called astrogliosis.
Okay.
When this response is prolonged or severe, these glia end up causing further damage to these neurons. It's sort of like they tire out. They can't keep up that extra burden of like holding the injury to that one area, and they end up forming scar tissue. They end up poisoning those neurons. So these glia end up sort of turning against the cells that they're supposed to protect, the neurons.
Okay.
And this is a problem. It's obviously, I mentioned that there's some injury before this, so it's not the initial condition, but this gliosis is present throughout these types of brain injuries, whether it's Parkinson's, Alzheimer's, ALS, traumatic brain injury, stroke, the list goes on and on, where you see this abnormal behavior of glial cells surrounding neurons and causing this toxicity. So for the last 4 years, I spent time trying to remediate the behavior of these glial cells, of a special type of glial cells called astrocytes, one of the most common glial cells, and sort of trying to push these astrocytes to stop misbehaving in a model of brain injury. So to kind of get these astrocytes to go back to protecting neurons and establishing healthy space in the brain rather than dumping out toxic chemicals, creating kind of a poisonous environment for neurons.
So what kind of toxic chemicals are they dumping out?
The main one I focused on was glutamate. Like you mentioned before, monosodium glutamate is a type. This glutamate is actually a neurotransmitter. So we have glutamate being produced by our brain cells naturally at small quantities just to send messages between brain cells. Okay. So if you're moving around, I'm talking, there's glutamate being released from neurons to other neurons, and that's causing the signaling. That's allowing me to function. But if this glutamate were to pile up around neurons at much higher levels than these neurons need, what happens is these neurons get overstimulated. They get overexcited and they just go into a frenzy, and that becomes toxic. It causes a lot of cell damage.
So why would glutamate congregate?
Well, that's a really interesting question. It's actually a chicken and egg question.
Okay. Crack that egg, Indrani.
Right. So we don't know exactly what comes first. There's glutamate that's normally being released. Now, these glia, the astrocytes I was talking about, part of their job is to be taking up this glutamate. Sort of, if the neuron lets out a certain amount of glutamate and the next neuron picks up a small amount, there's an astrocyte in the middle that's waiting to kind of suck up all the extra glutamate.
Ah, okay.
And to sort of process that into another compound called glutamine, and then sort of continue the cycle between neurons and astrocytes. Glutamate, glutamine, glutamate, glutamine. So there's a whole like recycling process that goes on via
okay.
astrocytes.
Wow. So all right, let me back up. We're getting a little heady. And I just want to kind of recap. No, don't apologize.
Please don't. Yeah.
I'm loving this. All right. So just to recap, and correct me if I'm wrong in any of this. So the brain is injured, either in many different ways, as we talked about. And what happens is these glia, which I talk about glia as being the support for neurons. These glia congregate around the injured neurons to protect them, right? And they kind of encapsulate them, if that's the right word.
Right. They seal off that area, or they try to seal off that area.
Okay.
There we go.
So, any inflammatory process would do, right?
Right. That is part of the inflammatory process.
Right. There we go. Okay. So that happens. But then what happens is glutamate seems to pool in these areas, which glutamate is an excitatory neurotransmitter, meaning that it excites the neurons and it can overstimulate the neurons basically to death. Right. And so what and this glutamate ends up just pooling around the neurons, because these glial cells that are surrounding the neurons are, as you say, misbehaving.
Right. Right. So
how are they misbehaving? What should they be doing?
They should be taking up that glutamate.
Okay,
and
so
in many cases, it seems they aren't.
Okay, so taking up glutamate, what healthy astrocytes do is they take up glutamate and then turn it into glutamine and useful compounds for the brain and body.
Right.
But they're misbehaving, so now they're not uptaking the glutamate.
Right. And there are other problems kind of in the middle. But the problem I studied most in depth was this accumulation of glutamate, because it's so potently toxic.
Tell us more about that.
It's the overstimulation. In a healthy brain, there's a very low level of glutamate between the synapses of neurons. And any disturbance to that it's one of the big things. Like I said, there's a chicken and egg story here. Like we can't exactly say one thing causes the other. But this in particular has the potential to lead to a lot of other cellular stress. We call it cellular stress.
Okay.
All sorts of parts of the cell that are damaged as a result of this overstimulation.
So if the astrocytes are overwhelmed, unable to fulfill their role of uptaking glutamate,
it makes the whole injury so much worse.
Okay, so then you have found a way to remediate this.
Right.
Please
So-
explain.
Before I get into the part where I remediated the issue, I want to talk a bit about how I figured out what the issue was. 'Cause
Oh,
the
yes.
truth is, we actually don't have a very good understanding of this glutamate piling up and the inflammatory process. Like, we know a lot about the two things piecemeal, but nobody can really fully agree where they intersect. So I kind of isolated this injury condition, and I looked at these astrocytes carefully when they were going through this inflammatory process, this gliosis.
Okay.
And
This is
what I
fascinating.
found was that the protein that was responsible, in my case, for taking up that glutamate piling around the cells
Yes.
moved away from the outside of the cell to the inside of the cell. So you literally have a protein that's in the wrong location and therefore can't do its job. Studies on brain injury are so evasive
Of
because we
course.
can't narrow down exactly what's going wrong. Everybody has a different impression. And I guess it's something we all have to work towards. So on my end, this was what I found.
Okay.
And what I did to fix it was I went about and created this healthy signaling complex from one's previous study of literature. I created this healthy signaling complex to give to these astrocytes to convince these inflamed astrocytes that nothing was really going wrong.
Okay.
And what this complex did was it pushed these astrocytes to make more of this transporter protein, the protein that moves away from the outside of the cell,
Okay.
as well as to increase the recycling of that glutamate.
So you're increasing the amount
I
of
increased the amount of glutamate that the astrocytes were able to take in from their surroundings.
Mm-hmm.
Okay. Okay.
So let me kind of recap what was just said there. So let me see, where do I start with this?
I know, the chicken or the egg.
All right, the chicken or the egg. We have the astrocytes not taking up glutamate. Glutamate's pooling, and then you find a protein that is what usually uptakes the glutamate is in the wrong location, and we don't know why this is the case.
I have
But
no
it's
clue.
the case. But it's
Just
the
a
case.
shout out to the audience here. If anybody potentially knows why a protein would move away from the cell membrane in a case like this, please reach out to me.
Yes.
Thank you.
Yes. Or you can contact Adventures in Brain Injury, and we will reach out as well.
That would
Yeah.
be much appreciated. Thank
Cool.
you.
Let's figure this out.
Many minds. Yes, many minds make for great work.
Definitely.
That's right. All right. So now, from previous research, you're looking at health. Explain that again. From previous research, you're looking at what healthy astrocytes are doing.
Healthy neurons are doing. So this is the part
Oh.
where it gets funky. Let's go back to the first thing I said. When neurons are injured, they send out a cry for help to their surroundings, right?
Sure.
That happens through unhealthy cell-to-cell signaling. So these neurons are calling to the astrocytes, help me, I'm injured.
Right.
What I basically did was the flip side of that. This complex that I isolated, it was found to be made by healthy neurons sent to astrocytes,
In a time of trauma.
not in a time of
No.
in a time of
Okay.
health.
Okay.
So I kind of flipped that, or what I believe I did on a conceptual level is I tried to flip that message the astrocytes were receiving to one of, oh no, everything's fine, I'm healthy.
Okay.
Nice. So basically, because they would be like, hey, I'm not healthy, come help out. And then they would come.
And that's when
And
the
then
freak
it
be
would-
process happens.
It would almost be
freak out
like an
process.
I love that. The freak out process. It'd almost be like an autoimmune deal where it just continues to
Unabated. Yeah.
immune response is triggered, but then it continues to be triggered and it's not
Right.
being turned off. So
Yep.
you're like, hey, let's tell this thing to stop. Let's tell it that we're
Chill
okay
out, man.
now.
Yeah.
Chill out. Come on
Yeah.
now.
So would you call
all right,
this?
So how'd
Yeah.
you
Go
do
on.
this?
Would I call this what?
Would you call this healthy signaling complex an anti-inflammatory
I don't know.
complex? Okay, so
It's actually
I
a
love-
very complicated complex.
I I love
of
this.
sorts.
This is amazing. Okay, we're on the edge of cutting neurological research. Indrani, back to you.
So what this complex really is, I guess, this goes into two levels of very new research that, honestly, I have to say it's almost like black magic. Because I was taking a huge leap of faith trying to find this thing. It's sort of like a needle in a haystack. You don't know really what it's going to do. So my complex had two parts to it.
The thing you're searching for is a protein that will uptake the glutamate.
That's what I'm trying to repair. So when I'm trying to remediate these unhealthy astrocytes, I'm trying to repair their use of that protein.
Okay.
Right.
So
and you're doing that by sending out healthy signaling to the astrocytes.
Yep. That's how it turned out, at least.
Okay. So explain what happens here.
So what I have is a part of that complex. It has two pieces to it. The first is the inner piece, the part that I think is having this effect of increasing that protein that'll take in that glutamate. It's called a microRNA. It's just a short piece of genetic material that functions in the cell to kind of chop up proteins as they're being made. And in this particular case, we don't know how this microRNA is increasing the production of this protein that I was interested in.
Okay.
But that was the effect. We found, it was previously shown that this microRNA would increase levels of this protein. Nobody knows how.
Huh, okay.
So that's another interesting thing.
More research is needed.
Yeah, definitely.
Yes.
So there's, on one hand, this molecule that I think is going to be therapeutic to my unhealthy astrocytes because I think it's going to increase the production and the maintenance of this critical protein that's going to take up this excess glutamate from the background.
Okay.
And the second part to this research, that complex, is the vehicle, the delivery vehicle of this microRNA.
Yeah, how does it get there?
Exosome. It's basically, if anybody has heard of a vesicle cells release these kind of bubbles of fluid and fat to surrounding cells as a way of communicating. An exosome is a tiny, tiny, tiny vesicle. It's on the smallest order we're aware of. It's really small. And what makes exosomes a great delivery vehicle, especially for small things like this microRNA, is that exosomes can cross the blood-brain barrier.
Right.
So the microRNA, it's then packaged into these tiny little vesicles, which are then delivered to signal the cell to create more of the uptake protein. How am I doing?
Very well.
All right.
I mean, it took me way longer than it took you, so I'm a little.
Yeah, but you're leading the way, Indrani. You've already done the hard work.
No, it took. Yeah, it was. It took me a long time to figure out what a cell was, so I'll give you that much.
Well, and now we can't keep up with you. You're amazing.
Well, hopefully our audience.
I've been kind of least.
I hope that our audience is getting some of it also. But yeah, it is. It's pretty in depth and
all of it is a conspiracy theory. All of it. There's no other way to see this.
What the neurons are conspiring against us?
Just the neurons and the astrocytes, and the interaction, the proteins moving away from the membrane, the restoration of the healthy signal. It's actually a murder mystery.
Keeps you up at night. Up at night. All right, so let's keep on going. So you find a way to go through the exosome, which actually, let's back up. What are exosomes?
They are just carrying vesicles.
So there's little vesicles that, what do? Connect cells?
In the normal functioning body, they serve as signals from one cell to another.
Okay.
So we
are they actually connected?
Connected? No, they're sort of like little bubbles that pop out from one cell and go to another.
Okay.
Like imagine like a bubble of like fluid that's sort of like cheering off little chunks that are like then flying out to cells in the surrounding. That's sort of what exosomes are.
Awesome. Little bullets.
Little
bullets of information.
It's like sort of passing notes between friends, you know.
Nice.
They're signals in our body, and
so right, these are messengers that are like, hey, create more proteins, or well, that's what the microRNA is doing. Create more of this kind of protein. But these exosomes carry other information as well.
Right. They carry all sorts of things, and we're just beginning to get into the heart of what these exosomes carry, what they mean for our bodies, like for different diseases, and how they sort of, because these exosomes are not always healthy. Sometimes they can cause harm.
Right.
Because, like I said, there's a distinction between a healthy signal and a harmful signal that I think most of my work was at least alluding to, if not using entirely.
And a lot of it has to do with timing, which is what I got from your work as well. Is like, all right, this is healthy at first, and then it gets crazy, and it starts having a little freak out session, as you said. So okay, so all right, might be getting into things we don't need. I don't want to like leave anybody's head spinning too much. So let me try to go over this one more time, super quick. So okay, brain's injured, ouch, and then the brain goes, hey, my brain hurts, and it signals to the astrocytes. The astrocytes go, cool, I'm gonna come over there, and we're gonna hold space for you. You know, they go in and they like close off the area so that other neurons don't get hurt, and then they, for some reason, start releasing glutamate or not uptaking glutamate, which they usually do. This glutamate is toxic to neurons, and neurons die as a result.
Of too much glutamate. Of
too much glutamate, yes, of overstimulation.
One of the results, yeah.
Okay, so you go in, you're like, well, the problem is we're not uptaking glutamate. Why aren't we uptaking glutamate? Well, because apparently when something's unhealthy, these astrocytes come to the area and they're closing off the area and not uptaking the glutamate because it's an injured area. Well, what if we told these astrocytes that everything's cool, and we send out a message that the neurons are healthy so that they can start taking up glutamate again, right? And that's where we're at right now. Yep. So now now did it work? What happened?
Right. So once I gave these astrocytes that healthy message with that exosome, these astrocytes started taking up far more glutamate from their surroundings. Now, the real acid test. So these astrocytes have like picked up the pace again. They're once again taking up glutamate from the surroundings. So basically the area around these astrocytes I thought would be less toxic to neurons. And lo and behold, when I put these neurons into this less toxic environment around these astrocytes that I'd treated, the neurons had a better survival rate. And not only did they have a better survival rate, these neurons, when I took pictures of them, they were growing more branches between each other.
You're
showing
kidding.
That these neurons were actually functioning better.
No. Nice. An improvement.
Right, an improvement.
Like beyond the baseline, an improvement beyond what you saw before.
I think it's comparable to what I saw before.
Okay. All right. Okay. Wow. And that was within a very short time period. Injury, treatment, resolution.
Right. Now I did all of this in like a petri dish of cells.
Of course, yes,
the time frame is definitely sort of an altered time frame.
Sure.
But it was within like a day. Like obviously it's hard to control the time frame because I mean if my treatment runs out it might start to get bad again. But in the time frame I tested, there was an improvement. There's a better time frame. Yeah.
And that time
neurons.
frame was 1 day. Just let me get that time
I
frame.
believe it was 1 day. I did it.
Okay.
Yeah.
Okay.
Wow. So, all right, neurons are surviving. Woo!
Phenomenal.
Call
Recovering,
for
yes.
celebration now. And all right, granted, this is in a petri dish. Like, we haven't seen this in a real live. This is in
Anything,
vitro,
yeah.
not in vivo, right?
Yes, in vitro.
So in vitro in a petri dish, in vivo would be in a live,
Mouse,
living
probably.
being, right? Such as a mouse. So cool. That's amazing. And then the way you were getting this microRNA into the system, well, we kind of talked about with it being able to cross the blood-brain barrier, you could potentially inject or even possibly take dietarily the microRNA that signals this and could potentially ameliorate brain injury. But again, this is all research, and we're
That would
trying
be
to
a
figure
lovely.
out.
That's like a lovely concept of being able to have these exosomes be produced to a treatment. It's many years off.
Right.
Partially is research, partially is just understanding how the heck these exosomes work.
So everybody, let's get this woman some research funding and get her into a place, because this is amazing research, so powerful, and I would love to see what we can learn more from this. And
So, Kevin,
yeah,
should we reveal the secret
Yeah,
kind
let's
of
do
right
it.
about
So,
now?
as I've been talking to Indrani, like the last time we talked, I had to pinch myself a few times to be like, okay, this brilliant young lady is 17 years old. And I'm like, wow, like the potential of this person is just huge. And so I just, high school, amazing what you were finding and figuring out. And like, as we are having conversations about astrocytes and glia and neurons and microRNA signaling and all of this, I'm pinching myself. I'm like 17 years old.
Well,
So, yeah.
yes, you have many years, and you still have the opportunity to develop this further at college. And so I'm thinking that you're going to have quite a few offers from labs around the country, if not internationally. Where are you at right now, Indrani?
Well, over the summer, I'll be working at Regeneron Pharmaceuticals, my benefactor, I would guess, in the whole paying for college process. So I will be working with them over the summer on some pretty cutting edge neural research that they have. And it's really exciting. And during the school year, as I attend college, I'll be working at Boston Children's Hospital.
And which college have you chosen out of the many on?
I'll be attending Harvard.
Of course.
Congratulations.
That's wonderful.
Thank you so much.
That's really, really wonderful. Well, there are so many. Well, and hey, Kevin, there's a lot of really great functional medicine. We need to get this woman in touch with Dr. Datis Karazian, who is also doing some study at Harvard. And Indrani, I see a ripple effect. People are going to be so interested in your work, and as a vibrant young woman with a ton of energy, great ideas, and no formalized training. I remember when I was at university, the big joke was you take a particular program and you come out as a trained SEAL. You do what you're told, and you're going into this with your own ideas. And I really think that's going to be paramount to moving research in a new direction, and having perhaps some of the old guard put the textbook to the side and let Indrani Das write her own chapter, if not a tome.
Thank you. But yeah, it's really exciting to be involved in this, especially when there's so much I don't know. And I have to acknowledge my research mentor, a biology teacher at my school, for putting up with all those 990 words of garbage every other minute.
Well, let me just say, I mean, I remind my young child, you had to fall a lot before you walked. And clearly your mentor, would you like to name your mentor?
Yes, Mrs. Donna Leonardi.
Oh, beautiful. Beautiful. So she clearly saw that there was a very big transformative process going on, and she was letting you take all of those little baby steps that were clearly turning into great strides. And one of the things, Indrani, I have to say, you have already embodied in the experience I have in functional medicine and functional neurology with some very intelligent people, is the one phrase that they say, you already say, and that is, I don't know. When I laugh, but not a lot of people are willing to admit that. The ego comes before the honesty, and I think it's just insecurity and fear. However, my experience with allopathic medicine was that the doctor always wanted to be right, and there was no room to say I don't know. And when I found myself researching and querying and asking functional medicine doctors, they too had gotten to the point where they said, I can't figure it out. I don't know. And that turned my world around. And I think that's how I started. That's how I started interviewing and assessing practitioners as I needed them for my recovery, was whether or not I tripped over their ego on the way into the clinic. Because if they were able to say, I don't know, then I knew that there was a world of possibility, just like you. You're just opening this world to whatever might be possible. So tell us about that, that long time ago when you were 14, 15, how were you introduced to sciences and what caught your attention?
I actually started in a high school that is a magnet school in New Jersey, and I had access to some wonderful labs and equipment. And I sort of just had this time in my lab with my mentor where my only goal was to find a disease or disorder and just find a way to start tackling it on the cellular level. And it was this incredible freedom. I could go and set my own standards for my work. I could just read papers, read scientific literature, push myself to understand what was already being done. And I could sort of slowly, over thousands of hours, just carve my path through subject like this, like neurodegeneration.
Unbelievable. And honestly, wow. Calvin.
Yeah,
I'm speechless. You go ahead.
Speechless.
Please.
Yeah, it's as you were talking about, Michelle, so a theme that we often talk about on this show is how being in a place of curiosity is so much more useful to patients and to doctors and to science than being in a place of arrogance.
Yes,
and,
yes.
I often talk about how science knows that science doesn't know it all. The people who think they know it all, they don't know what science knows, which is that it doesn't know it all, right? And so, I see so much. I love the research you've done. Your focus on detail. Your thinking outside of the box, and just being very open and explorative, and it's just a beautiful thing. And as somebody commented on a post I made with you that featured you, they said a child will lead us. And I was like,
well, too late for that. In a couple months, I'll have turned
it's true.
adult world. So.
Well, we have already established that you are beyond your childhood years. This afternoon, I'm going to be touring a lab here in Calgary. It's mitogen advanced diagnostics lab. And I am going to have to introduce you to the director of that lab, just because he is one of the humblest men I've ever met. And he has every reason to stand on a pedestal. So yes, I'm going to do that today.
So much. I can't tell you how much that means to me.
Yeah, I've got to say. Yeah. Okay, we have taken quite a bit of research time.
Yeah.
This laboratory researcher cabinet.
Very true. I do want to say one last thing, and that is that I would absolutely love to see you get what you need in order to advance this research forward. So reaching out to my audience, if anybody can help this brilliant young lady elevate what she is doing to elevate our understanding of brain injury and brain injury treatment and what can be done in safe and effective ways, and more research is needed to find out. So just want to reach out and put that out there. You can contact me, and I'm in touch with Indrani, and I would love to hear from all of you. So thank you so much. Anything else that you'd like to add, Indrani?
Thank you for having me. It was an honor to be able to talk to you. And honestly, I have to say, I was ecstatic when you reached out to me over Facebook. Both of you are an inspiration for me. And it's just, I would say that my motivation at the most fundamental standpoint is to be able to preserve the sort of spirit and humanity the both of you embody. It bothers me that brain injury takes away from the identity that people create for themselves.
Well said.
Seeing two people like you who went through that and were able to rebuild not just the same identity, but something so meaningful for so many people. I mean, it impresses upon me why we would take up this yoke and try to fight back the loss of identity that's caused by all these forms of brain injury.
Oh, I think there's a circle of inspiration forming here. Oh, Indrani, please keep in touch.
Thank you so much for that.
Was
that
just
was so beautiful.
Yeah,
Thank
very,
you.
very touching. So that's inspiration up here in Canada.
This is now the Committee of Mutual Admiration. So thank you guys so much.
Thank you.
Yeah, we appreciate it. Pleasure. Take care. Have a wonderful day.
You too. Thank you. Have a great weekend.
Bye-bye.
Bye.
All right, y'all. That's it for this episode of the Adventures in Brain Injury podcast. If you enjoyed this one and want to hear more like it, head over to iTunes, Stitcher, Google Play, anywhere else where podcasts are found. We're probably on there. And leave us a review. That helps us out a lot. And if you'd like to support us, you can go to adventuresinbraininjury.com/support, or you can go directly to Patreon/AIBI for Adventures in Brain Injury. Throw us a buck or two for each podcast. That helps us out a whole lot. If you'd like to sponsor us, head over to feedabrain.com/sponsor to see the sponsorship opportunities that we currently have going on. Thank you so much for your continued support of Adventures in Brain Injury, and we will see you next time. Adios.
Someone take me to a doctor.